Okay. So good afternoon, ladies and gentlemen. First of all, I would like to thank Professor Thomas Koenig and Professor Petra Ritter for organizing this workshop, which I'm very proud to be part of. So with my presentation, I'd like to show you a general overview about the problem of decimation of brain connectivity. And since some of you may be not familiar with this, I'll try to be general and to give you an overview of the general principles, in particular of a family of methods which can be used for the estimation of brain connectivity, and also to show you some practical considerations, some caveats and pitfalls or suggestions for practical applications of such methods. And I also try to underline some possible future perspectives. Thank you. Okay, so as I said, since not all of you may be familiar with this, with the brain connectivity study, I'll just give you a short introduction on why do we want to study brain connectivity. So as for the definitions, I'll give you soon, but in general, studying connectivity means trying to describe brain networks, the organization of brain areas and regions during specific tasks or even at rest. This organization can provide interesting insights into the mechanisms at the basis of some complex functions, for instance, cognitive functions, which are based on the interaction of large networks of our brain, but are also important and interesting when studying modifications in such brain organization resulting from, for instance, different pathological conditions. And finally, the brain organization and reorganization is at the basis of the phenomenon of cortical plasticity, which is in turn at the basis of learning and of the recovery of lost functions as a consequence of pathologies. And such recovery can be both spontaneous or induced by a rehabilitation or by a treatment. So it's particularly important to study how this reorganization happens in the brain. So we want to study brain connectivity, which is in general the information flow moving through different areas of our brain. So once we have a map of activity in different regions of our brain, we are interested to estimating this kind of network here. So a very general outline of the presentation. As I said, we are interested in studying such networks, but the very definition of what causality and connectivity is a problem itself. So we need to start by a definition of connectivity. So we start from here. Many different definitions of connectivity are possible. We may be interested in studying the existence of physical anatomical links between different regions of our brain. This is the anatomical connectivity, which is very interesting, very important. There are methods to study this kind of connectivity, but this is not the topic of this talk, because the existence of anatomical links does not imply necessarily that there is information moving through them. The study of this information moving through regions started by seeking for a kind of temporal correlation or coherence between the activity recorded in different cerebral sites. So correlation or coherence can tell us if there is a link between different regions, because if there is a temporal link, if there is a synchronization of the signals, probably there will be a kind of link between those regions. But still, this is not a circuit. So if you know that some regions are part of a circuit, and you know that they are linked, but you don't know the direction of that link, you don't know how what happens in one region can have implications on what is happening in other regions, you can tell that you are building a circuit. So to get closer to this ambitious goal, to modelize brain circuits generating the data we are experimentally observing, a third concept arose, which is the concept of effective connectivity. Here again, there are different possible definitions of effective connectivity. This one is the one I like best. So we aim to get as close as possible to the brain circuits generating our data. What we can do is to build the simplest brain circuit that would produce the same temporal relationship observed in our data. So, of course, this does not mean that the circuit we are building is the true circuit, but we are getting closer and closer to that. Still, if we want to, so this is not a measure, this is not a thing we can measure. Connectivity cannot be measured, we can estimate it. And to give this estimation, we need a model. So here, many possible ways are open in front of us. I will show you this one because it's one of the most simple, the most straightforward, and one of the most effective we have today. But other ways would also be possible. So the family, as I defined it at the beginning of the presentation, this family of methods for the estimation of brain connectivity all start from this definition, which was given by Norbert Wiener in 1956, which is the definition of causality in the statistical sense. So according to Wiener, if we have two simultaneously measured signals, simultaneously measured is important because, of course, we are seeking for something which is happening in time. If you can improve the prediction of the first signal by incorporating the information about the past of a second signal with respect to the prediction you can have just by using the information about the signal itself, then you can say that the second signal has a causal effect with respect to the first one, a causal effect in the statistical sense. So this is a very nice definition. It's very general, it's very precise, but still we cannot use it to compute the connectivity estimation. What is lacking is a model. So here it is the concept of prediction of a signal. There is a concept of past information influencing the current data we can record, but we don't have a model. The model was added to Wiener's definition by Clive Granger in 1969. So the definition of Granger causality is exactly the same as the Wiener's one, with the exception here of the use of a specific kind of modeling, the autoregressive modeling. So according to Wiener-Granger causality, we can say if we have two time series simultaneously recorded, if we can improve the prediction of one time series, for instance, B, by using the information about the past of another time series, A, with respect to the prediction of B just made on the basis of the past of B itself. So by using this information, we can improve the prediction of another signal. Then we can say that, in this case, A has a causal effect in the Wiener-Granger sense to B. So the most important contribution of this definition is that we have a model that we can use, and that we have, this is from Wiener's definition, we have the concept of directionality. Not a prevalent direction, but we have both directions, because you can do the same by reversing the two-time series, and you can seek for an influence directed from B to A, which is conceptually different from the influence directed from A to B. So you have two distinct contributions in the two directions. So to get very briefly inside this definition, this is ready to be applied. We just need two very simple models. One is the autoregressive linear model here, in which we consider an estimation of x at the time point n just as a linear combination of past samples of the time series itself. Of course, we have a prediction error, which is the residual of the model. And the second model is the bivariate autoregressive modeling, which is essentially the same, but here we have inserted in the linear regression for each of the two time series the past information about the other time series. So here we have other possibly different residuals of this model. And so we can just simply apply the definition by Wiener and Granger. And so we just need to define what does it mean to have an improvement in the linear estimation we are doing. And we can consider this improvement in the linear estimation as a decrease, a significant decrease of the prediction error. So here in this formula, which represents the Granger causality test, we can, for instance, direct it from Y to X. We can compare the variances of the residuals of the univariate model modelizing X on the basis of its past, and of the bivariate model modelizing X on the basis of its past and of Y's past. And we can have two possible outcomes. If the past information about the past of Y does not improve the prediction of X, then there should be no improvement, no reduction in the residual of the model. So these two terms are more or less the same. This is 1, and the Granger index is 0, so no causality. If by inserting the past of Y into the prediction of X, we can have an improvement of this prediction, then there should be a reduction of the variance of the bivariate model. And so here this is greater than this, and this is greater than 1, and we have something which is different from 0. So the Granger test, it's very simple, very straightforward, but carries many different important information. The existence or not of a directed causality between these two signals with a value which is proportional to the improvement we can get in the modeling by inserting the information about the past of one signal. So these are the advantages of this method. So it's very powerful. It was a kind of turning point in the history of the estimation of causality between signals, not only in neuroscience, but in general. It was developed for economics purposes and social sciences, and then it was lent to neuroscience. But still, this method shows immediately some very important limitations when we want to apply it to our questions, to our brain connectivity estimation. The first limitation is due to the hypothesis under which we can build these autoregressive models. So you cannot just take any kind of signal, any time series, and build an autoregressive model on them. You need the signals to verify some hypothesis. And the main hypothesis is that the signals should be stationary, at least in the weak sense. This is a very restrictive hypothesis for us because it means that we can analyze with these methods only, for instance, if we are dealing with neuroelectrical data, we can analyze only data during time windows in which signals are stationary. Some parts of our experiments are not good for this kind of analysis. And we completely lose any information about the transient of connectivity. So if we want to track changes in connectivity in time, for instance, as a consequence of a stimulation, this is not possible because this violates the hypothesis of stationarity, which is required by the model. And this is the first strong limitation. The second limitation was underlined by Granger himself in 1980. Because all this is true only if all the information, all the sources of activities in our problems are limited, are included in the two time series we are using for our modeling. So only in this case we can see that what we get from the Granger test is causality, is true causality. Otherwise we go into another kind of problem, which I will show you soon. So bivariate versus multivariate approach. Let's imagine that we have three sources of information in our problem. Of course, we can still use bivariate methods. We can select one of the sources as a seed and seek for all connectivity with all the other signals. Or if we are interested in building a circuit, an entire circuit involving all possible interactions between those regions, we can select all possible pairs in our dataset and apply this bivariate approach to each pair. What happens let's imagine a situation like this. There is one source influencing two sources which are not related. What happens when we apply the Granger test or any other bivariate method to this couple of signals here? Since these signals are both influenced by a third source which is not visible by the model here, there are some similarities between them. They are related in some way. There is a kind of correlation between them. And since the model just takes into account these two signals, it's impossible to prevent the method from finding this kind of false positives, false links, which are just due to the fact that the information is not available entirely to the modeling. So what we get when applying a bivariate approach, in particular the Granger causality, but as I said, this is very general, a general problem of all bivariate approaches, is that we get something which is different from the correct model. And this difference gets worse and worse with the dimension of our model. So there are simulation studies showing that you can get a huge amount of false positives by this approach. On the contrary, what is a multivariate approach? A multivariate approach, you have a multivariate approach when you include all the information about your system. So if it's a multi-channel system, if you have more than two sources of information in your problem, you put all these signals into a unique model. Of course, the model is much more complicated with respect to the bivariate one, but if you are able to estimate correctly such model, all the information is there simultaneously. And so, for instance, in the previous example, when you try to estimate connectivity between these three nodes, these three areas, here the model has the information about all of them together, and thus is able to distinguish the fact that the similarities between 1 and 2 are due to a common effect by 3, and not by a direct connection between them. So you can have more accurate estimation of these brain circuits by using multivariate approaches. Of course, the implications of this are that the model is much more complicated. This is a multivariate autoregressive model. If you look at it more closely, it's exactly the same that we had for the bivariate approach. But here we consider, in the estimation of each channel or each signal in our data set, the possible influence of the past signals of all the other past samples of all the other signals in the data set. And this is done for all the possible signals. So you estimate all the possible interactions between the signals in our data set. Another possibility which is given by this approach is to move from the time domain to the frequency domain. Of course, we can still keep working in the time domain, looking at the residuals and seeking for the information, the causality between all possible pairs in this data set. But if you are interested in the frequency domain, and if you are using EEG or MEG data, you should be interested in the frequency domain. You can also we can also use this same model to move from the time to the frequency domain. If you look at the multivariate autoregressive model in the frequency domain, what you have is a filter with n inputs and n outputs, and you have a matrix of you have a transfer matrix, which is this one. It's based on the parameters of your autoregressive model. Each of these functions puts in relationship one input to one output to of your filter. So one signal to another signal in your data set. And this is not a symmetrical matrix, which means that you have two different contributions when you look at influence directed from n to 1 or from 1 to n. And this is true for all the possible pairs. So the directionality is still there. So many different methods were built on the basis of this modeling and on the Granger causality concept. Here I reported these two because they are the two original ones, the first to appear. They are the directed transfer function and the partial directed coherence. In particular, there were some evolutions of these measures and of PDC in particular, including different normalization to improve the physiological interpretability of what we get. But despite the normalization, despite the kind of interpretation you can give of the number associated to this estimator, what both these estimators can return is a distribution, a description of how this Granger causality is distributed at different frequencies for all possible pairs of signals in your dataset for both directions between them. So you can build a network like this one, or a bigger one, of course, if you have more than 3 signals at different frequencies. Now, we have a method, we have a family of methods. They are very good for EEG and MEG data because of the properties of this data, because of their statistical properties. We have to decide to what kind of data we want to apply these methods. So the question is, sensor space or source space? Of course, you can apply these methods on scalp data, for instance, if we are talking about EEG. This is not forbidden, you can do that. I will show you also some results of the application of these methods on the scalp data. The main problem, of course, is still the same we have for all the other analyses we can perform on EEG, on scalp EEG. It's the problem of the spatial blur of the data. And the spatial blur means here that when we get a signal from this electrode, we are not just looking at what happened in the region just below the electrode, but probably we are capturing also information coming from different regions, and the opposite. So this information coming from a brain region can be spread over more than one electrode. This is bad for brain mapping, and this is even worse for brain connectivity, because when studying connectivity, we search for kind of correlation between the activity in different electrodes, and of course we may find a correlation which is just due to this effect. There are some mitigation of this problem. A first mitigation, for instance, is due to the fact that when you use Granger causality instead that, for instance, coherence or correlation, Granger causality, as I said, is based on a time lag between the two signals. So if this propagation is as it is, as we believe it is, instantaneous, this kind of correlation may not affect Granger causality, because Granger causality just detects an interaction with a time lag, not simultaneous, not a simultaneous interaction. So a possible mitigation is given by this fact. But still, it's very difficult to give a physiological interpretation of the networks we can get on the scalp, because we cannot attribute what we see on an electrode to specific brain regions. So, of course, we may profit of all the methods developed during the years to back project the activity we record on the scalp on, in the source space. So, for instance, to achieve the maximum accuracy, we can use all the information we have also from other modalities, for instance, from MRI, to build realistic models of the structures of the head, which are interposed between the generators in the brain and the sensors on the scalp, to build realistic models which can be used to increase the accuracy of the reconstruction we can have in the source space. If this information is not available, we can use average models. If you use them correctly, you can still have a quite good improvement with respect to the sensor space. And then you can compute connectivity among data related to this estimation. So you can compute connectivity between the activity estimated in different brain regions. So this is what we would like to have in all cases. There are some caveats, some pitfalls, or some points that should be took in mind when doing this. A first caveat is related to the fact that we are used to perform some procedures, some pre-processing on our EEG or MEG data, some kind of filtering, artifact remotion, ICA, linear inverse problem solution, and so on. Some of these procedures may be good for mapping, but not for connectivity. Because for mapping, if we induce or if we remove some correlation between the data, this may not be too bad if it does not alter the relation, the reciprocal amplitude of different parts of our mapping. But for connectivity studies, this can be a disaster. So each step that we take, we must ask if it's inducing or removing a correlation which is in the data, because this could alter significantly the connectivity patterns we have. So this is a point to keep in mind. A point which is good, that I already told you, is that instantaneous correlation should not affect Granger-based methods. So when thinking of this kind of problem, we should also take in mind that instantaneous correlation is not that bad. And a very important point when dealing with this, and especially when trying to move these connectivity applications to the clinical side, is that in case of lesions of the brain or lesions of the structures of the head, for instance, when we have a traumatic brain injury or if we have a stroke or any other pathology that can alter the anatomical structure of the head, we cannot use average models, of course. We need the specific models to be built for the specific patient. This may be cumbersome, may be difficult to have in clinical situations, and I will show you some example later. Finally, when moving to the source space, we usually need larger dimensional model, because when moving to the source space, then on the scalp, what we can have, the information we can have is limited. So we can reduce our analysis to a limited number of electrodes. No more than 30, otherwise you just have redundant information. But if you move to the source space, usually you need a larger model to include all the possible sources of your problem. So this is also, this should also be took in mind. And to link to this last part about the model dimension, this part, I would like to address this part together with the problem of stationarity, because there is one method that can provide a solution to both problems. So, as I said, the second major limitation of Granger causality and all MVAR or AR-based connectivity methods is the hypothesis of stationarity, which is required on the data. This is very restrictive, as I said. To overcome this limitation, we try to move from conventional MVAR models to adaptive models. Okay, a first way you can follow is to select short time windows in which you can consider your data as approximately stationary. You can do that. You can have small time windows and sliding windows moving through your experiment. And this is a possible way to overcome this limitation. Of course, the time resolution is not that good. Another possible way is to consider MVAR models with time-dependent parameters, which means that you perform an adaptive fitting in which the model parameters are updated sample by sample to follow your data. So the requirement of stationarity of the data is no longer there. You do not need the data to be stationary anymore, and you can follow changes in connectivity with a good temporal resolution. There are two main possible approaches to this. The first one is based on the recursive least squares, and it was introduced in 2003 by the group of Professor Herbert Witte in Jena. The second one was developed in cooperation with my group and the group of Professor Witte in 2010, and it's based on a general linear Kalman filter. The second method, the general linear Kalman filter, as I will show you later, is also able to deal with the problem of dimensionality. So when moving to time-varying connectivity, what we get, don't be scared by this picture. This is an example of application of a time-varying connectivity estimation to 12 brain regions in the source space. So what we get from these methods is for each pair of areas in your dataset, for each direction between them, a description of a distribution in time, this is time in samples, and frequency, this is frequency in hertz, of Granger causality in a full multivariate approach. So you have a lot of information here, you need to summarize them. What you can do, for instance, is to select a frequency band to average the activity, the connectivity in that band, and to have a time course of connectivity directed from one region to the other in time. This was a combined foot-lip movement in a group of patients with a spinal cord injury and in a group of control subjects. So for instance, you can see during the movement preparation how connectivity directed from one region to another evolves in time for each specific frequency band. Or you can, if you want to have a general look at the entire network, entire circuit, you can take snapshots or you can make a video of time evolving connectivity networks during the movement preparation. Like here, for example, for one patient and one control subject during the movement preparation. As I said, this was achieved by means of the estimation of adaptive MVAR through a general linear common filter, which has another advantage with respect to other methods. And the advantage is that it allows an accurate estimation of these big models, even if you don't have that much data. So it's very robust, and it can work even if you have a reasonable amount of data, which is not true for other methods, because the model is too complex, and it usually needs a lot of data to be estimated. But with this approach, you can have fairly good estimations on large dimension models, even if you have an amount of data which is reasonable to obtain. Now, as I said, the problem, this is very shortly just to show you some applications. Such methods, if you apply them correctly, and if you have, if you can obtain a good estimation of these models, can provide a plethora of information to you, because you have an information about the existence, the direction, and the intensity of connectivity in the time frequency domain. So you have a lot of information, and you may need some possible way to summarize such results, also to give an interpretation to such results, especially, as I said, if you don't have the possibility to go to the source space. So the interpretation of this data is also a crucial part. Very, very shortly, what we can do is to summarize the properties of these networks by means of a number of indices that are derived from the graph theory. Graph theory is a mathematical framework which is used in many different contexts. And its major contribution to connectivity studies is that they provide a simple way to summarize the properties of the network. From the most simple ones, like for instance, the density of a network, how dense it is, how many links are there in the network, to most complicated ones, like the kind of structure inside the network, if it is efficient or not, if the kind of organization it has in itself, the prevalent role of specific regions in the network, and so on. We can define many, many different indices according to our scientific question. So if we are interested to a specific role of a region, we can define an index to measure that role, for instance. And what we get from our time-varying approach is time-varying graph indices. So we don't have just a snapshot of a network with some numbers describing its properties. We have a network evolving in time with some indices which are evolving in time too, and describe how the properties of the network evolve in time. So here I don't know how to make the video start. Okay, thank you. So this is a very simple foot movement to which we can, from which we can compute, from the EEG data related to this, we can compute multivariate connectivity networks estimation, like you see here. And this is how the network evolves in time during the movement. Of course, it's very difficult to derive any conclusion from this, but we can compute some indices, quantifying, for instance, the general properties of the network, how the network is organized, how the network is, how is the energy consume of the network, some very general properties of the network, or local properties like the role of specific areas in the network. And all these indices are evolving in time, like our networks. So this is an example of how we can extract information from our network. And this is particularly important, as I said, when we are not in our ideal conditions. For instance, if we want to apply, this is an example, I will provide you 3 examples of application of these methods to real situations. The first example is related to the evaluation of the effects of motor rehabilitation. So in this experiment, the aim of the experiment was to evaluate if, you know, that motor imagery elicit an activation in our cortex, which is quite similar to the actual movement. So it has been hypothesized that motor imagery can help improving the motor rehabilitation. But still, it seems not to work. So the hypothesis above this study was that if we support motor imagery by a brain-computer interface system, which provides the subject a feedback of his motor imagery, this can boost motor imagery itself and thus the recovery of movements. So we performed this study with 28 post-stroke patients, which were divided into groups, one group performing just motor imagery and the other performing motor imagery and BCI, supported by BCI. And the results, from the point of view of the clinical outcome, indicated that there was a better functional outcome in the BCI group with respect to the control group. This was this was reflected by a significant increase in the results in a Flugmeyer scale, which is a clinical scale to measure the functionality, in this case of the upper limb in particular. So this was obtained from the clinical point of view. We were interested to understand what was happening from the point of view of the brain, how the brain networks were reorganized after this training. And here we had the problem that the MRIs of these patients were not available. It was not possible to have, to analyze this data in the source space with the methods that we usually use. And so we were limited to the scalp level. As I said, this gave you some limitations from the point of view of the interpretation. Here, the graph indices I briefly introduced to you can help you. Because, for instance, since we know that some properties of the networks which are disrupted in stroke are related to the connections between the two hemispheres, the interhemisphere connections, we computed an index called interhemispheric connectivity, measuring the amount of significant connections between the two hemispheres which were significantly improved after the training with respect to the screening performed before the training. And so by this comparison of pre versus post-training connectivity, even if we cannot have a clear description of the brain circuits underlying this improvement in the task, we can have a hint about that on the basis of a significant improvement of this interhemispheric connectivity index. So, of course, the best way would be still to have the possibility to move to the source space. But if you can't, there are some ways to give an interpretation of the results you get on the scalp level. The same was done here. This was a different, still a group of post-stroke patients, but it was a cognitive rehabilitation. And we wanted to see what the effect of this rehabilitation was on memory functions for the patients. And here again, we didn't have the possibility to move to the source space, so we stayed on the scalp's surface, we computed connectivity, and we extracted some indices related to local properties, but local properties of the network. For instance, the anterior density, which means the prevalence of connectivity in the anterior part of the scalp with respect to the rest of the network, or the left temporal degree, which is the involvement of left temporal electrodes, because we knew from this working memory task that some processes were involving, related to this memory task, were involving the left temporal regions and the frontal regions. And in this way, we could get a statistical analysis showing that in the patients which were responders to the treatment, there was a significant increase of these indices, which was not there for the patients which were non-responders. So in some way, we could get to some indices that are accurate and stable enough to capture differences between responders and non-responders patients, and to in some way describe roughly what is happening in their brain reorganization. Again, we would prefer to have it in the source space, of course. This is a correlation we got between these indices and the behavioral data reaction times and percentage of errors. Before moving to this last very short example, the main message of the two examples I gave you about stroke patients is that we need a method to go beyond the analysis at the scalp level, also for subjects which cannot be studied by means of average models. So having the individual MRIs of each individual patient can be not possible, or making all the computation behind that may be not so possible in clinical applications. So possible perspectives which would be very useful for this kind of clinical applications may include some kind of methods to get a reasonably accurate estimation in the source space, also for subjects which have lesions. This is a main important point which I would like to take to your attention. Then the last application is a very funny one, but also very exciting one for me. It's one of my topics of research, and it's the estimation of connectivity related to social interaction in multi-subjects recordings. So I'll be quite short on this, and I'll tell you soon why, but shortly, when studying social interaction, there are proofs that considering information coming simultaneously from different subjects recorded during their interaction can produce much more information about the neural basis of their interaction than studying them separately. So what we can do is to record couples, pairs, or groups of subjects during interaction tasks, and to compute connectivity by means of methods that are based on the ones I showed you in this presentation, to seek for kind of functional, of course this cannot be a physical, but it's a statistical connectivity. It's a causality in the statistical sense between the activity in parts of the brain of different subjects when they interact. For instance, in this experiment, which was related to a game in which subjects could decide to cooperate together to defect each other or to punish each other, which is the tit-for-tat condition. On 52 subjects, we found that the individualistic condition, which was the defect, showed a much less connection or no connection, no statistically significant connections between the activity in the two brains, while on the contrary, we had a significant number of links between the two brains in the interaction parts of the task. This was also confirmed by a classification in which we could discriminate the individualistic parts of the task from the cooperative or interactive parts, just on the basis of the properties of these intersubject networks, with an accuracy which was up to 91% in the theta band. So this is very short, because if you're interested to this is short advertisement. If you're interested to this, I have a poster tomorrow and an oral session on Thursday dealing with this kind of intersubject connectivity estimation. So if you're interested, you can join me at the poster or at the oral session, and we can discuss about this. So with this last application, I would like to conclude this presentation. I would like to acknowledge the contribution to the results I showed you here of my group and different groups we cooperate with. And of course, I thank you all for your kind attention, and I'm here for questions. Thank you very much.
Thank you. It was a very nice talk. So, because you said that true causality can only be measured when you take into account all the signals in the process, so it made me think, how do you think the choice of the number of sources will actually affect the causality results? Because we have to find a compromise between the number of sources, and the computational time. And the approach that we currently use is to divide the brain in 82 regions of interest and take only the solution point that is the centroid. But this reduces a lot the model. So how do you think this affects actually the connectivity results?
Yes, it for sure has an effect. So this is the crucial point in this kind of estimation. Ideally, you should include all the sources of information, of course, to be sure that that you are not introducing false positives. One thing that you can do to because of course you will have false positives, because it's not, as you said, you need a compromise between how much information you can use for your model and what you can achieve due to computational and time constraint. Also, for many reasons, you cannot just include all the sources into your model. One thing that you can do is to use different conditions to, in some way, eliminate some false positives. What I mean is that if you know that some possible effects of false positives due to the lack of some sources of your problem can be there in your experimental condition, but also in a baseline condition that you can design, if you compute connectivity in both conditions, you will have the same effect, which is not related to your task, but it's just due to this kind of confound. And by statistically comparing these two conditions, you can eliminate part of this problem. Of course, not all the problem, because if some effects are really related to your task, they cannot be eliminated. But then you should carefully consider which sources introduce in your model. Because if you introduce all the relevant sources in your model, relevant for your task, other effects which are just due to a baseline activity can be removed by this kind of statistical comparison between tasks, task and baseline.
I would have another question, if that's okay. So there's a lot of discussion about these bivariate versus multivariate methods, but for example, imaginary coherence, it's very widely used. And so what do you think about the use of this bivariate method versus multivariate Granger causality?
What I think is that you should use, in my opinion, multivariate methods anytime you can. So if you have enough data to perform, because you need more data to perform this multivariate approach, because the model is more complex. If you don't have enough data, you don't have good results, so it's useless. And it's better to switch to bivariate methods, even if you know, but you should know that you will get a lot of false positives. If you have enough data to move to a multivariate approach, I think you can only benefit from that. Because you have more precise, there are many studies showing that you have more precise circuits and more, for instance, you can have completely, you can have properties of the network related to small wordness, to high efficiency, which are not there, which are just due to these false positives. If you apply, for instance, graph theory to a network built by means of bivariate method. So you, as I said, you can use bivariate methods if you cannot use multivariate ones, but you have to know that some properties of the network may be significantly affected by that.
Thank you.
Okay, thank you very much. Okay, no, please, please. Sorry, Christoph.
I wanted to discuss a bit, if you have a little bit of time, the stationarity issue that you mentioned there.
Yes.
The problem that we are not allowed to use these things in non-stationary data sets. For example, one of the things where it is often applied is epileptic activity, intracranial epileptic spikes, which is a problem of stationarity, I think. Do you have any suggestions of how we can, get rid of that, come over, bypass this problem, and still do some sort of connectivity analysis in this case?
Yes, this time-varying connectivity based on these adaptive models can be a possible solution, especially the Kalman filter-based one. Because the Kalman filter-based methods do not require multi-trial recordings, which of course cannot be used for epilepsy applications. And they do work very well even on single trials. Not very well, of course, but they still give some reasonable results also on single trials. So I think that this adaptive Kalman filter-based approach could be useful for these epileptic applications. Okay. Thank you very much for your attention.
Thank you very much for your talk.
Okay, another question.
Yes, sorry. It's just.
I'm very happy. I just don't want to
it's just a very short follow-up to this
one.
Yes.
Yes.
You mentioned earlier that there's also an alternative possibility to reduce the problem, so that you simply use small sliding windows.
Yes.
How small do you think we can go with EEG?
It depends on the dimension of your model.
Yeah.
If your model is very big, you cannot just have very short time windows unless you have many trials.
And let's assume I did my MOLED wavelets in a way that I optimized it for the time resolution. Would 50 milliseconds be okay? Or is...
In general, 50 minutes?
50 milliseconds.
50 milliseconds.
Minutes would be amazing.
50 milliseconds, I've never dared.
Okay.
I went up to 200, 250 for short time windows. Of course, in general, it's a question of trade-off between your time window, the signal-to-noise ratio you have, and do you have multi-trial recordings, I guess? Yes. If you have many trials, you can try.
Okay.
But you really need a lot of trials.
Thank you very much.
You're welcome. Okay, so can I go now? Thank you very much.